Hello, how are you?
Hello.
Good. Sorry for the small delay. We'll just head into the presentation today. Does anybody has any questions so far about anything? Maybe about the presentations that follow. If anything is unclear, we can maybe discuss it now because the presentations are next week, right? So I think I don't know how many of you have already put the topics in the group. I know one group has, but the others. Excellent answer.
I sent the paper via email. So
Okay. Did you send?
via email?
Did you already send it, or you said you will send?
No, I sent it. We discussed it over Viber.
Oh yeah, yeah. But that's one, and then so who is with you in the group?
Leila, Tarin, and Sarah.
Okay, and the other group was who sent me? I have it in Viber. Yasmina. Okay. So what group is that?
Mi, Berina, Jasmina, Đorđe I Igor.
Okay, and then the other group is Armin, and what's the third group left?
One group is me, Samina, Ilham, and Adna, and I put the link of our paper in on the drive link we
Okay.
used before.
Okay, good. And the fourth group is Irnes,
Jest.
Aisha, Lamia.
Jest.
Jeste vi stavili?
Ja nisam ništa stavio, ne znam za njih dvije, jesu što uradili.
Pa morate zajedno.
Nismo se ono dogovarali, pošto ja lično nisam imao vremena, tako da stavit ćemo to ako nije problem za vikend.
Dobro, pa imate samo 7 dana vremena da završite tu.
Ma, završit ćemo.
Dobro.
Vrijedni smo mi.
Dobro. Normalno. No problem. So we'll then have it in the format as we discussed. So if you have any questions on how to structure it, if you want to send it beforehand to me so I can have a look, we can also do that, and then I can give you some advices, because I told you all the stuff we talked from the first presentation. Hopefully you took notes, and we'll be judging your presentation skills on the notes we took last time during the presentations. Okay, good. So I'll just share my screen. So we're going to talk a little bit more in depth about immunogenomics. So we're going to talk about, as you can imagine, autoimmunity, but also about hypersensitivities of any kind, genomic structures of immune cells, and then particularities about genetic recombination or genomic recombination in order to obtain T and B cell receptor specificities, so-called VDJ recombinations. So just have to kind of refresh our memory of the stuff we already talked several times about. So what are autoimmune diseases? And they're a unique category of diseases being characterized by an immune response to antigenic components of the host itself. So autoantigens. This is kind of a outdated, I would say, hypothesis, because autoantigens are not necessarily needed to be present in order to have an autoimmune reaction. Right? There's several other. So there's T and B cell mediated autoimmunity. And can anybody now? I didn't have immunology, right? So I can't really ask you these questions. But understanding that B cell autoimmunity would be mediated by autoantibodies generated by B cells, right? And impaired activity of cytotoxic T cells, CD8 positive, would be T cell mediated. So the destruction of the tissues in organ-specific disease, for example, would be mediated by cytotoxic T cells rather than autoantibodies. And as you can see, there is a substantial overlap between environmental factor, genetic predisposition, and immune regulation. So historical autoimmune diseases were considered rare, but through rigorous epidemiologic studies have now been shown to be somewhere between 3 to 5, depending on the population, somewhere 7 to 9. The more north you go, the more autoimmunity is present. There's a north-south divide, not only autoimmunity, but in hypersensitivities in general. So we can see then this prevalence north-south divide is affecting Western societies mostly. And the stuff we know about population genetics and the similarities of basically everyone. So the more or less the genetic heterogeneity of populations from in Eurasia is not that big, let's say, in comparison when you look at Africa. So why would you, for example, assume that Western societies would have an increased in autoimmunity? I think there are two possible explanations for it, but I want to see if any of you can come up with an idea why there would be a west-east divide or north-south divide in autoimmune diseases. Somebody needs to answer.
Maybe because of our diet and the food we eat.
Yeah,
Because
that,
a lot of like the trend of the fast foods and things like that.
there could be very well a possibility. So lifestyle in general, that could be category, right? Lifestyle pollution could be also another thing, which is, or at least was present more in industrialized societies. Stress as a trigger for diseases could also be. In general, industrialization makes perfect sense. But another thing is access to healthcare also, where just more people are tested for diseases. Hence, statistics are just reflecting more and more the real situation, which was always present in society. So I would assume there is a combination of both. In this case here, you have to know that autoimmune disease affect mostly women. The ratio in most autoimmune disorders is 3 to 1. Do you know maybe that could be? So what there could be also two answers to this possibly.
We go through more stress.
I would say so. I don't know, possibly, but also there's we know that autoimmunity, for example, is a hyperreactive immune system, and androgenic hormones like steroids, anabolic steroids, or androgenic hormones in general, like testosterone, are immune dampening, for example, right? So immune dampen mean that it's immune suppressive, and more testosterone would mean more immune suppression. So women could technically have a less suppressed immune system because of the lack of testosterone, for example. And that's a very neat and nice hypothesis, I would say. But also that, at least in the West, right, women are much more likely to go to the doctor and to describe skin conditions or like psoriasis, for example, or pain in general. So just statistically speaking, there is a higher possibility of women reporting disease status. So all of those confounding factors, like there's partially biology to blame, most definitely, right? But also socioeconomic and societal developments cannot be neglected in this case. So it begs the question, is it genetic, so autoimmunity, or environmental factor, or both? At this point, I think we know that any disease is neither environmental or genetic. It's all a combination of both of those, except monogenic diseases, right, where you're just born. But then also that's a kind of environment, right? Maybe your mother was drinking or your father was 70 or mine was 45. So it's also an environmental factor to it. So or timing in general. So this is, I think this becomes more and more a silly question. Is it genetic or nurture versus nurture? I think only unserious people really look in that and that explanation to begin with. So there nothing is just nurture or I mean, even this environmental aspect, it does not affect a vacuum, right? Or an ether, it affects genes. And then those genes are affected through transcription or translation in any form, right? In the 3 steps of the dogma. So I think it's a little bit too reductionistic and actually does not do any good in trying to explain anything in a deeper sense. But when you think about genetics, you can also see that we have people who are 11% of left-handed people are affected and have autoimmune disease, while only 4% of right-handed people have autoimmune disease. This is just, I think, a funny correlation. So there's, I think, no real studies behind this, trying to understand any kind of biology behind it. But the surface understanding could also be that we know that left-handed people are more creative and more. There's a different, I guess, in utero hormone structure of maybe the mother or who knows what kind of effects, but there is a slight connection between those 2 dots. So creativity and autoimmunity, and most left-handed people are women, so it's also kind of a huge combination. Anyhow, so this is just kind of the world spread of autoimmune diseases worldwide. So you can see that you have systemic lupus, which has the highest prevalence in the US, for example, in comparison to other developed nations. While and then you have multiple sclerosis evenly distributed. So there's also a geographic divide in some of those diseases and the prevalence of the population suffering from the disease and the male to female structure. And for example, where was India? For example, we see here India in some places. Where was it? Yes, here, for example, like he would see more males in India, which is has most definitely do something with testing. And also here, we see more males having the disease. Wait. Here's also Japan. So most of those differences which are observed here could be epidemiological in nature, just people reporting more or less or not being able to access healthcare in general, especially in India and those places where women's rights and those kind of things are not that pronounced. But we know that all those diseases are kind of manifested in a spectrum. So we just say autoimmunity or autoimmune diseases, but in general, we mean all those states from mild reactivity to the self to extreme reactivity to the self, split into different organs. But the kind of basic mechanism, as we know at this point, is the same. So it's either, I mean, in any case, it's your body attacking itself, just a way of transmitting those attacks to oneself is different. We said the T cell or B cell mediated. And we know that autoantibodies play a huge role in rheumatoid arthritis, where autoantibodies can be fired in synovial fluid from the knees and joints. While other diseases, we know there have no antibody signature. But it's obviously clinically very relevant to split those disorders and different diseases, organ-specific or systemic. But thinking about just, I would say, conceptually about what autoimmunity is and how the immune system functions, you will see that there's a gradients in all those conditions. You know, people with multiple sclerosis, for example, who live more or less normal lives with stronger or lower relapse situations than you have people who die from multiple sclerosis because the um, myelin sheets are degraded in the brain and the neurons are open and you basically can't live and functionally normal. But then there are people with lifestyle choices, diet, THC consumption, for example, mitigate tremors and lots of stuff caused by multiple sclerosis. So it's most definitely a spectrum of how it affects people who are suffering, but also as a family of diseases, there is a gradient in transition between the systemic and organ specific. So one needs to keep that in mind also, I think, when researching those things. And we also spoke about those internal and external factors, but just to sum up, this would be this kind of interplay. And we now know that, for example, that multiple sclerosis, there's a groundbreaking science paper last year who actually found that Epstein-Barr infection, it's most definitely a trigger for multiple sclerosis in people who have it, which is also a little bit funny because you think about it. Epstein-Barr and herpes virus and VARS and all this stuff is um, so common in the population, right? So prevalent. So it's very hard to uh, correct for it. But so the study actually looked into frozen blood samples from, I think it was the Army biobank, which this screening looked for. So it's kind of interesting to see. It's kind of verified because this hypothesis was of viral triggers was actually coined many years ago, but I think just the last nail in the coffin of that hypothesis to be true was just last year's paper in Science. So, can anybody? I know you didn't have immunology, but does anybody can tell me the kind of main organs of the immune system and what they do, for example? I mean, they're obviously written here on the graph, right? But does anybody know what, for example, the thymus does?
Does it produce T cells?
Yes. So it's so immature lymphocyte basically leave the bone marrow and find their way to the thymus, where they're then educated and become mature T cells, basically. So it is an interesting structure, and it's much bigger in children, right? Because then those T cells needs to be trained, and you have like this thymus, which is this kind of it's a cortex and a medulla, and all of those MHC molecules are there exposing the cells to it, and there's positive and negative selection, and then you have your CD4 and CD8 cells leaving the thymuses, then mature already exposed T lymphocytes. Another organ which sometimes not referred to immune organ is the liver, right? So because we know that the liver synthesizes proteins and complement system, and it contains also a large number of phagocytic cells, which increase bacteria in the blood, and then they pass through the liver, so they get rid of them basically. Then we have the bone marrow. Bone marrow, can anybody tell me what the bone marrow does? What kind of cells are in the bone marrow?
B cells.
So the bone marrow is a location where all cells of the immune system begin their development from primitive stem cells, basically. So that's how they develop. Then we have the tonsils. See? So then we have the tonsils, which is basically a lymph node. Nothing more. It's just called slightly different, but also like the spleen, for example. The spleen is also just a huge lymph node. But do we know what lymph nodes are? So it's a place where basically B and T cells congregate, right? So um, we have all them all over the body, so they store there, and when there's action and infection, we have this immune system. You learn which kind of is kind of the non-adaptive one, which is the kind of first line, and then they figure out, oh, there's something going on, and then we need to call the adaptive immune system. And then, yeah, I think without you having a course, it does make sense to try to squeeze this into 2 sentences, but it's very important because, as I told you, the immune system is the second most important, or the most complex organ system behind the brain. The interactions are countless. Only the brain rivals it because the brain is exponential complexity. So many layers, right? You could, for example, aim to understand the immune system at one point in time fully, but I think that we will never really understand the brain, at least not with the technology which we have now, right? At least the level of consciousness and perceiving reality and those things, but not to bother with philosophy, no. So, and then we have obviously the blood, which is kind of circulating all the stuff. Without the blood, none of this works. And then we have cells of the immune system, which would be really good for you to know. So we have the granulocyte, mast cells, monocytes, which basically develop into macrophages, which are super important. Then you have M1 and M2 macrophages, dendritic cells, natural killer cells, B cells, and then you have the subsets of T cells, CD8 positive, the cytotoxic ones, CD4, the helper, which has a complete subsets of those itself, Th1, 2, and 17, and then T regulatory cells. They all, and now actually new studies even show that we have cytotoxic CD4 cells. It's just getting more and more complicated. So with granulocytes, so they have those kind of cells here until this point, and then you have the T cells. So then we have I think 4 types of hypersensitive reactions. So type 1 are allergies. So an allergy is an allergic reaction provoked by a re-exposure to a specific antigen, referred also as an allergen. Type 1 is distinct from type 2 and hypersensitivities. Type 2 hypersensitivities is an antibody-dependent process in which specific antibodies bind to antigens, resulting in tissue damage or destruction, so like Graves' disease, and so back and so forth, acute rheumatoid fever. Then we have type 3 hypersensitivities when there is a combination of immune complexes, antigen-antibody complex that have not been adequately cleared from the immune system, which is like systemic lupus, those aggregates in the brain. And then type 4 or T-cell mediated diseases, which are diabetes mellitus, the insulin-independent one, rheumatoid arthritis, multiple sclerosis, and inflammatory bowel disease. So this would be 4 types of hypersensitivities. And the most common autoimmune disorders are rheumatoid arthritis, Hashimoto's thyroiditis, celiac disease, Graves' disease, and diabetes mellitus type 1. And there you can see what organs they affect. So the concept of immunological tolerance is defined as an ability of the immune system to prevent itself from targeting self-molecules, cells, or tissues. So key concepts of self-tolerance. So we have central tolerance, that's in the thymus and bone marrow, which play a key role in shaping immune system homeostasis, which means the balance basically. The thymus developing lymphocytes undergo positive selection in the cortex before maturing and entering the circulation. In an otherwise healthy host, lymphocytes with potential reactivity against self-peptides are negatively selected and deleted in the thymic medulla. So the body tries already to get rid of those before they come out into bloodstream. Importantly, after exiting the thymus, mature T cells are subject to secondary selection, peripheral tolerance, by which the majority of self-reactive T cells are deleted or rendered anergic, meaning they can't do stuff. In addition, if a mature B cell specs surface IgM that recognized ubiquitous surface antigens, they are eliminated by a process known as clonal deletion or clonal anergy. And then we have autoreactive B cells that can escape and then produce all those antibodies which hurt you. So, and then we have this peripheral energy, T regulatory cells, superfluid energy is this stuff which I just said. So basically, when B cells express surface IgM, and then they are emitted by this process, so the body knows that they're kind of redundant and not needed, but sometimes they also escape this process, and then you have the generation of autoantibodies. Then Tregs, so T regulatory cells, are a specific type of CD4 positive cells which act as immune dampening. So their presence, for example, in tumor tissue has a worse prognosis because it blocks the immune system of doing stuff. So if you see a lot of Tregs, you know the immune system is suppressed, basically. And then we have homeostasis produced by cytokines, chemokines, and other cognate receptors. So the kind of stuff which is produced by antigen-producing cells. And this is just how it somehow looks. So we have the bone marrow, where B and T cells are maturing. So even though the most strict control by central and peripheral tolerance, a small number of autoreactive T and B cells leak out into periphery in normal individuals. However, they will remain harmless unless their genetic predisposition to break a tolerance and environment triggers to disease. You can see we have the bone marrow and the thymus, which we talked about, and then those mechanisms of tolerance, but then again, autoantibodies, autoreactive T cells and B cells, and then regardless in which direction, we have the pathological autoimmunity and physiological autoimmunity, which is a transient apparent long-term resistance, no tissue damage, maintain homeostasis. This happens all the time, right? So in your body, there's nothing spectacular to it, but this is the stuff where one needs to be on immunosuppressive drugs for a lot of years, if not ever. And there's a concept of so-called molecular mimicry, the theoretical possibility that sequencing similars between foreign and cell peptides are sufficient to result in the cross-activation of autoreactive T or B cells by pathogen-derived peptides. And this is basically how it looks. So you once get infected by a disease, and then your body produces your own proteins and shows them on these MHC molecules. But then for some reason, everybody thinks that your own protein is looking like the protein of this Epstein-Barr stuff because they have a half similar amino acid composition and it fits here and it causes a reaction. So just now, fun question. Do you know what this disease is? Can you tell me that? Do you have
Artosa?
It's rheumatoid arthritis, right? I don't know if this said, I think it's in our language arthritis, but I'm not sure. Rheumatoid arthritis. Yes, so this is the symptoms of rheumatoid arthritis. So morning stiffness in your hands lasting more than half an hour, simultaneous symmetrical joint swelling, not released by rest, fever, weight loss, fatigue, anemia, lymph nodes enlarged, and then we have nodules forming in your lymph nodes. And then we have diagnosis of RA. You see how the x-rays look. This looks very painful. So no single test to specify rheumatoid arthritis except autoantibody testing, verification of individual joints, scans, synovial membrane biopsies, antibody tests, and inflammatory markers. And genetic-wise, now we come into third parts, there are more than 100 known risk loci shown by genomic studies to be associated with the disease, but the only very strong finding is HLA-DRB1 associations across cohorts. Does anybody know what this disease is?
Celiac.
Yes, exactly. It's a celiac, and this is a disease triggered and sustained by the ingestion of gluten, or not really gluten, but it's actually gliadin, a component of gluten, in generally predisposed individuals. And cases causes an inflammatory damage of the mucosa of the small intestine, with resulting malabsorption of nutrients. So in order to get the disease, you need to inherit either DQ2 or DQ8, but they're not sufficient to develop the disease. So absence of HLA-DQ2, DQ8 has a high negative predictive value. So let's see. Yes. So celiac disease, contrary to popular belief, there are no gluten sensitivities, right? You either can eat gluten or can't eat gluten. That's very simple. I know a lot of people now watching on television, they tell you can do this genetic test and then you're sensitive, you have shouldn't eat gluten, you shouldn't. If you're AB positive blood type, you should eat this and don't eat chicken. It's all nonsense. Yeah, if anybody tells you that stuff to be real, I mean, that's really like homeopathy. I mean, it's even worse to some degree, I guess, because it pretends to be scientific. I know a lot of private firms in Sarajevo where you can test your genes for see what food you should eat best. And, that doesn't really work. I guess in that sense, psychologically it works. You give the money for a test, and then a guy tells you what to eat, but in general you cut calories, so you feel much better. So yeah, anyhow. And this is the prevalence celiac disease in combination with other autoimmune disease, which is quite specific for this disease, and also a lot of Down syndrome people, and other physical and mental retardations also often have celiac disease, but also diseases which do not prone severely mentally or physically impaired or retarded phenotype, like Turner syndrome in females, where girls never become reproductively viable and stay small in stature and look like children basically, but they're otherwise more or less completely fine. It's 90% of them have celiac disease too, so that's a kind of interesting observation. And here you can see how. So this is the small intestine here, and this is a normal small intestine, and this is under the microscope. You see how those valves are nice and clear. So this is partial atrophy. So you see in the electron microscope how those nice absorby thingies look after the destruction of the immune system due to gliadin. And this is total atrophy, right? Those people are bleeding into their stomach cavities, and inner bleeding and blood in the stool, and it's extremely disgusting and painful. But it's actually reversible, which is funny. So as soon as you stop eating gluten, this becomes this again, because nevertheless, those people have much higher instances of colon cancer, and yeah, which is a little bit also crazy, right? When you think about it, a disease which destroys your intestinal cells develops cancer and then forgets how to destroy the same cells, giving you cancer. So this is just ridiculous how the body works, but it is what it is. So can anybody tell me what this disease is, for example?
MS?
Yes, that's multiple sclerosis, right? So a lot of people have those tremors because of the damaged myelin, which actually, as you can see here, which is a fat layer of the cells. And then electricity is conducted through the insulation layers, and then make your movement when you think and do stuff and you see. But when this myelin is damaged, then those nerves fire in an arbitrary way, and you just, yeah, it's not a very nice condition to have. So MS involves an immune-mediated process in which an abnormal response of the body's immune system is directed against the central nervous system, which is made up of brain, spinal cord, and optic nerves. The exact antigen or target the immune cells are sensitized to attack remains unknown, while MS is considered by many experts to be an immune-mediated rather than an autoimmune disease. So this is the damaged myelin, as you can see, and the exposed fiber of the nerve. And this is a Schwann cell, how it should look. Right? It's just nerve fiber. Looks like a bit like an optic fiber cable. Right? So the genetic factors about autoimmune diseases, those are the important genes that regulate the development of autoimmunity, are located within the MHC, which you had for your exam, and hopefully read thoroughly through it. MHC have got critical role in maturation of T cells and induction of IR. MHC2 genes are directly responsible for autoantigen processing and presentation. The structure of AG binding group, antigen binding group, will determine the specific antigen will trigger an autoimmune response. And the famous HLA complex, a gene complex encoding the MHC proteins in humans, a set of cell surface proteins essential for the acquired immune system to recognize foreign molecules and retroviruses, which are in turn determines histocompatibility. Function binds antigen derived from pathogens and displays them on the cell surface for recognition by the appropriate T cell. And this is nomenclature of HLA genes. You see the HLA, then we have the gene ABC, DRB, DQ, whatever. This is separated, and then the allele group, specific HLA protein, synonymous DNA substitution. Then we have our used to show difference in the non-coding region, and then a suffix denotes the changes in the expression levels. So it's a very, very thorough annotation of the gene. So we have 3 classes of HLA molecules, 2 classical and 1 non-classical, called HLA class 2. So class 1 is consistent with HLA A, B, and C genes expressed on all nucleated cells. And what are all nucleated cells? So what does that mean? What cells are they not expressed? So all nucleated cells means all cells that have a nucleus. Which cell does not have a nucleus?
Blood cells.
Exactly, right. So, and they present endogenous antigen to T cytotoxic T cells, right? And they have the class 2 molecules, which are HLA, DR, DQ, and DP, expressed on B lymphocytes, activated T lymphocytes, monocytes, macrophages, and Dixon cells. And how are those cells collectively called? By any chance, does anybody know? They're called antigen presenting cells, and you can memorize this. It's going to be easy for a course in immunology. So, and they present exogenous antigens to helper T cells in order to recruit those guys here. So those take it up, right? Decompose the foreign bacteria, virus, or whatever, and then express the peptide fragments here in the class 2. Then the CD4 sees it, and the CD4 doesn't do shit except calling the killer cells, and then the killer cells go and kill, given the information which this CD4 provided, because it induces clonal expansion in those given the antigen that it encounters, and now all those clones which can kill the bacteria or the virus just grow. And that's why you need 2-3 weeks when you get vaccinated or when you are infected. It's always like incubation 7 days, then you're sick for 7 days, and then you're getting better for 7 days, because this process needs time. And that's why when you have memory cells, then it's easier, then this stuff goes much faster. And this is the genetic composition, as we tell, class 1, class 2, and this is class 3. And we know that there's more than 250 loci there. And this is how those genes, when they code for something, this is how they look. So this is class 1, and you can see this is coded, this whole part is coded by one gene, it's A, O, B, or C. And it is beta-2 microglobulin, which is a supporting molecule. So this is the cell, right? And this is this molecule. It's not a receptor, so always remember MHC is not a receptor, it's a molecule. It's connected to the cell surface here, and class 1 is coded by one gene, and the structural support coded by a completely different gene, which has nothing to do with antigen presentation. But the class 2 is coded by 2 different genes, and that's why it can bind a lot of different peptide fragments from different diseases. So technically speaking, given that you have 8 billion people on the planet, we can actually survive with no problems any given bacterial or viral infection from anywhere. So cytotoxic T cells is how it looks. A recognized complex of viral peptide with MHC class 1 and kills infected cells. T helper cells recognize complex of bacterial peptides with MHC class 2 and activate macrophages. Helper T cells recognize complex of antigens and activate B cells. So genetics and disease. So we know that we have 3 billion base pairs and roughly 20,000 protein coding genes. And we know that 99% of genetic information is identical in any two given individuals. Now, human phenotypic variation lies in the remaining one. What does this sentence mean when we say all human phenotypic variation lies in the remaining 0.1%? Can anybody elaborate on that sentence, for example? You can think about it a little bit.
All of the differences found between humans are only due to that 0.1% of genetic change, and everything else basically is the same.
Exactly. It means from hair color to disease predisposition. Just a second. My boss sent me a message. I need to answer this. Just a second. I need to make a call. Okay. Good. Sorry for. This variation can be split into structural variation and simple nucleotide variation. So we have SNPs, genes, and haplotypes. So this is just refreshing memory. A SNP can be described as a change in a single nucleotide that occurs in a specific location in the human genome. A SNP is not a mutation. We talked about this. The combination of two SNP alleles that a person has on a particular pair of homologous chromosomes is called a genotype. Haplotypes is a sequence of more alleles along a single chromosome, and linkages equilibrium refers to the non-random association of alleles. I'll send you this presentation, by the way, of alleles, so 2 or more oocytes in the general population. So this is the genotype, right? So this is one mommy and this is daddy, right? And this is the haplotype. So we can genotype those SNPs and we measure genetic variation of SNPs between individuals, whole genome genotyping, microarrays, disease susceptibility, phenotype traits, testing for allele frequency shifts between affected and unaffected individuals. And this is just a different approach. Microarrays, which, I think we talked about it during my presentation last week, but they're very potent and very energy or very DNA saving platforms with only 200 nanograms of DNA in order to give you up to several million of variation across the human genome. So, and the genotyping workflow is amplification DNAs, amplified PCR, fragmentation, precipitation, and suspension, hybridization. And I think those steps you should know quite well. So, and this is the hybridization via an array. The array contains millions of tiny spherical beads embedded in an array surface. Each bead is coated with hundreds or thousands of copies of short DNA sequences specific for a locus of interest, right? Here we see 3 beads on the Illumina array. Each of the beads have different probe sequences. For simplicity, only 2 probes are shown. Specificity is ensured by the length of the probe, buffer conditions, and elevated temperatures. And The probes are enzymatically extended with a single labeled base to disseminate between genotypes. Homozygotes will have identical label bonds, while heterozygotes will have mixture of labels. The DNA template is then removed, leaving only labeled probes on the beads. And this is how you can see it. So the second component of this is the process is staining, adding a fluorine signal to the probe. And not to bother you, I think this is just redundant, and I'll show you how it looks. So this is basically genotype not called, and heterozygous and homozygous, and then you read it up, and you know for what gene it was done, and voila, you know what the SNP is on a given position. And this is a genetic association study, where basically you use this technology which we talked about previously, people with the condition, people without the condition, and then we compare the genomic information, and then we identified disease overlapping. And this is the stuff we, I think, showed you, and this is how our plot looks. And next time we will actually try to generate on our lab those kind of plots. Not next time. No, next time we need to finish. This stuff from last time, but after New Year's, for example. But until when is your semester actually?
It's, what do you mean? It's until February.
Is it until February? So we have whole of January, right? Still
Yes,
lectures.
the exam preparation week is like the last week in January, something like that.
Okay, good. So, and this is how we represent genome association wide studies and data with the SNPs, every SNP represented by one dot, and transform p-value on the y-axis, and the x-axis we have the chromosome. Overlapping genetics in autoimmune disorders. So on the genetic level, the HLA gene complex is a well-known contributor to autoimmune disease. Most of the identified autoimmune diseases have been shown with the class 2 variant of HLA genes. And then we have another interesting group of very polymorphic genetic genes in immunogenomics or immunogenetics, which is killer cell immunoglobulin-like receptors, which is a cluster of approximately 15 genes on human chromosome 19. KIR expression is stochastic, basically meaning random. Linear correlation of gene copy number and NK cell expression KIR. It's assumed that there is only a single ancestral KIR receptor, which from all those 15 developed. And we see a lot of duplication, mutation, and recombination due to non-allelic homologous recombination. And it has a cognate HLA ligands, which is also kind of mysterious when you think about it, because this is on chromosome 19, MHC is on chromosome 6, but they somehow work in tandem, even though their inheritance is completely independent, which is still blowing my mind how that works. But yeah, it's just interesting. NK cell repertoire relate to KIR haplotypes, so we have a coevolution with HLA, and this is how you nomenclature KIR. And I won't bother you with this. It's too long, but basically you have a number of extracellular IG-like domains, length of their cytoplasmic tail, the number of different KIR receptors with the same number of extracellular domains and length of cytoplasmic tail. And when you add this all together, you get KIR2DL1, for example, where all of this is representing the colors. I think we can leave it now. In case of recognition, and then the genetics of that will take a little bit more time. Good. Could you follow? I guess not that much, but at least is some of the things does make sense. I know it's more a crash course in immunology also, together with the genomic part, and it's very unfortunate that you didn't have immunology at this point, because my course is very immune dependent, so to say, because my background is like that, right? So, but yeah, so you could follow more or less. I mean, any
Yes, it's perfectly fine.
okay, good, my friends. So tomorrow we have our coding session, And then I'm there on next Thursday at 3 o'clock. We'll meet. Maybe 3:15, right? I'm landing at 2 o'clock. We'll meet at 3:15, 3:30 in a room. I think I send you already, but if not, I'll send you in what room we meet for the first presentations. And then we meet on Friday on 9 o'clock in the morning and then take it from there. Is that good?
Yes.
Okay. Thank you. See you then tomorrow. Bye.
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 Bye.